IGB, the Institute for Genetic Biology, is basically built around the idea of trying to do really kind of cutting edge research in a specific areas of science, themes of science. And if you look at the building, it's kind of hard to see from the outside, but the building is basically cut up into very big laboratories, these cooperative research environments. And they focus on everything from host microbe interactions to, I'm trying to think of a few other ones. Our group, which is bioinformatics or computational biology, biocomplexity, looking at the evolutionary origins of modern organisms, looking at stem cells. So we have a regeneration biology theme, synthetic biology, and so on. So we, as an institute, really focus on specific areas, and the labs are sort of divided up into those areas. As an institute, you can also refocus research depending on whether things change. I mean, over every 5 years, they kind of do a review of how research is going, and they'll look at the themes and see if they need to refocus direction. That happened in this last few years. And a few of the themes have changed basically research direction. But it's a very interesting environment to work in. You get a huge range and diversity of people doing research. In this one building, we have our group that does computational biology. We have Jean, who does honeybee research. We have Rachel, who we'll talk in a bit, who works on microbial ecology, but in a very broad range, in an evolutionary context. We have, Rachel, I don't know if you have anybody else that you can add to that? I mean, it's a pretty broad building, research-wise. Geology, yeah, I mean, it's just a huge, huge range of researchers in this one building. I don't know if there's anything more to add, basically, than that. So a lot of this research really hinges on sequencing, to segue into that. So I'm going to go over basically where sort of the history of DNA sequencing and technologies. I'm really only going to focus on two because I don't have a lot of time to go into a lot of detail. But a brief history of sequencing technologies, to go over how sequencing used to be performed up until the mid-aughts, about 2005, 2006, and then the current applications of what's called next generation sequencing, what we actually use today, and then a couple of real cases where next generation sequencing has had something of an impact that you may have seen actually in the news, and these are good things and bad things. So we can kind of discuss or openly discuss some of the implications of the so-called personal genomics era, where anybody can have sequence data generated. So the first major breakthroughs for sequencing DNA were basically in the mid-1970s. There were two major papers that generated two major methods for sequencing DNA. The first one that really came out was called the Maxim-Gilbert method, and the second one was the Sanger method. So Walter Gilbert and Fred Sanger are the two researchers shown here. These are the actual papers, and you'll note that they came out in '77. One was in February, one was in December. Obviously, there was a lot of work going in this direction. Sanger's method proved to be the one that everyone pretty much went with, because if you look at Gilbert's method, it had lots of nasty chemicals in it. People didn't like working with those in labs, so they actually went with Fred's method. Both of these guys won the Nobel Prize in 1980 for this work. So very important catalyst in molecular biology for actually promoting getting genetic information in a manner that labs could get to and access. So I'm going to focus on Sanger sequencing. I'm just going to go over some of the basic detail of it. The original method for Sanger used radioactive labels. So you would get this, you've probably seen these DNA ladders. you could visualize DNA fragments. You would run 4 reactions, one for each of the terminators that you would use in the reaction, and I'll talk about what that means in a second. But later this changed over to a fluorescent system, so you can see that these have different colors, and the colors are based on the base. This is run on a gel-based system, usually separating everything by electrophoresis. So I'm going to do a quick demo. I think hopefully this will work. All right. So this actually is a nice animation that came from the Wellcome Trust. Sanger was actually, I guess, honored with the idea of, or with a center of his own name called the Sanger Center in the UK. It's now called the Sanger Institute. And actually a lot of these animations can be found, if you ever want to go and look for things to use in classes, they have lots of wonderful materials at the Wellcome Trust. So he actually started developing this method in '75. He didn't publish it until '77. It's known as the dideoxy or chain termination method, and you'll see why. So basically, you're going to actually run a number of reactions in one well. So you're going to have terminators that are going to actually halt the extension of DNA at a particular base. Okay, those terminators are going to be based on the bases of DNA, A, T, G, and C. And these copies are going to vary in length, so you can separate these out using basically common electrophoretic methods. These have been modified so you can generate lots and lots of data. So the reaction is initiated generally at either end of a target DNA. The key thing is that you have to have a single-stranded DNA. So you'll see if we unwind this, it'll go to a single strand. And then you have to have a primer that will bind to a known region of the sequence. The polymerase is going to extend. It's going to add nucleotides and extend. And at a certain point, it's going to hit a terminator nucleotide, in this case a T, and stop. Same thing will happen with other nucleotides. So you get basically a reaction where you have stops at various positions, and at each one of those positions where it stops, that nucleotide is labeled. These are all occurring in a single well, okay? So this basically goes into a nice sequencer. This is an ABI. And you'll see this is one of the reactions in the well. The strands will go through. This is a capillary tube, but it actually has basically a gel matrix in it, so it still works like electrophoresis. It separates them through this gel matrix based on size, just like you're separating a gel, an agarose gel, but this is a much more refined gel. It can separate based on base pair resolution. As these pass through, this is a laser, and it will actually illuminate or fluoresce each of these tags, and you'll have a different color pop up based on the base at that position. Okay, and these are all separated based on size, so you're basically reading these off as they're coming off the gel. So you can read from the bottom up. This is the base closest to the primer. This is the base furthest. And so you can just read the bases as you're going along based on the color itself. So again, he got the Nobel Prize for this. The downside of this method is cost. So to do a Sanger-based sequencing, and actually the original human genome was sequenced in this manner, it costs $3.3 billion, okay, for one genome. So it's going to be very hard if you were wanting to actually do this on a scale where you want to do this on a personal level, sequence your genome or your genome to look for the causes of disease. It's just not feasible using this technology. $3.3 billion for each one. You could probably get the cost down to around 300 million, sure. But it's not really feasible. So really, there was a huge push if you're going to go towards the era where you can sequence and make it economically feasible to sequence individual genomes. There was a very big push towards what are called next generation sequencing technologies. A lot of these technologies are developed. Yes?
I just had a question about the Sanger sequencing, because you said that they need a primer in the beginning.
Yeah.
But how, if you're sequencing a genome and you don't know what's in there, what would you use for a primer?
So you would generally have these cloned into a known vector. Most of the time you have these in a plasmid, or you could amplify these using PCR. And PCR has primers on both ends that are known. Okay. So you have the sequences based on that.
When they were first doing the sequencing, didn't they sort of distribute as such the genome across the country? So one lab here had this and one lab there had that. And so it's really a big group of.
Yeah, so this is basically the center, which was kind of the amalgam of all of the different sequencing centers, but they basically handed out money to Baylor, to Washington University, to Utah, to the Broad Institute. So it had all these large sequencing centers that acted as the main hubs for doing this work. What you're seeing these days is that is slowly dissipating because labs now can do this on their own. It's actually kind of interesting now. So the big push was to these next generation sequencing technologies. The challenge was to get to generate sequence in a way that was cheap. The sort of gold standard, or the thing to aim for, was the so-called $1,000 genome. So medicine, for instance, there are some medications that can be covered if they're $1,000 on a monthly basis even. Insurance companies would have no problem in paying for those things if it actually meant increasing the lifespan of an individual. So it's not out of the realm of possibility that an insurance company or somebody would pay to actually have their genome sequenced if it were $1,000. That's sort of the gold standard to go with. So the idea was to try to promote or push towards that goal. The one I'm going to talk about is Illumina, but the real key one I think that started this, there was Roche developed the 454 machine, which is called a pyrosequencer. You'll see a couple of slides in here that include that information. But Illumina basically came out as the technology that really won out, and you'll see why. So this is a current version of the Roche sequencer. Alvaro Hernandez, who operates the sequencing center, actually gave me these slides. So you'll have an idea. You'll probably see it'll be a little salesy, sorry. But you'll get an idea of what it can do. So this machine, if you use it in two different modes, can generate 50 to 600 gigabases of DNA in a day. So gigabase is 1 gigabase is 1 billion bases. The human genome is 3 billion bases, okay? And in general, if you want to do any good work, you need a pretty decent level of coverage for the human genome. You still need about 50x based on this technology. That's 150 gigabases. Well, that's 600. So you could generate 4 human genomes in about 11 days. Okay? If you wanted to even be a little faster, you could probably generate 1 human genome in 2 days. So that pushed it from the original scale of a decade and a half to 2 days. So I'm going to go over this, and I really want people to, if they get lost, to raise their hand, because Illumina is a little different in the way it works. It's still got a bit of Sanger in the way it works as well. You'll see it uses terminators, but it's a little bit more involved. So there are basically 4 steps. There's the library preparation step, the cluster generation step, the sequencing step, and data analysis. These last two steps actually occur pretty much at the same time. It's ongoing. Yep.
Just a quick question. The Sanger method.
Yeah.
It's a probability, right? It's a probability in the sense that the polymerase will fall off and you will get all of the bases.
It doesn't fall off. The key thing is that it actually is a chemical reaction. It literally stops. It terminates at a particular position.
It's a probability in the sense that the di-terminator gets incorporated at some rate.
Yes. Yeah. So that's something I don't go into a lot of detail on because that sometimes confuses people. There's a mix of actual nucleotides plus some terminator nucleotides in there. So you get some read-through, but you terminate at some positions. This is a little different in that you always terminate, but the terminator is set up in a way that you can unblock it. Okay? All right, so to give you an idea of what this is all run on, this is a typical high-seq slide or a set of lanes. You can barely tell, but there are about 8 different lanes in this one run. Each of those lanes can hold 400 million sequences. Okay, about 200 million spots, but you can do what's called a paired end run and get 400 billion sequences. I should clarify that. But it's a lot of data. It's a huge amount of data. All right, so the way sequencing is done now is kind of interesting. You basically, it's a service. It always has been something of a service. A lot of people used to do this in their own labs, but these instruments cost about $500,000 a piece. It's actually not terribly expensive to do this on your own, though. You basically would say, here's my DNA sample, Alvaro, please sequence this for me. And what Alvaro will do is he will generate what are called libraries of sequences. He takes your DNA sample and he fragments it, he chops it up. He does a little bit of repair, but then he adds these adapters to the end. So you'll see these are Y adapters that are added to the ends here. Now, these are necessary for the next step, okay? Because what you're going to do is you're going to take this DNA with these adapters on the end, and you're going to run it on these lanes. So if you're looking at the lane, you're looking at the bottom part of this lane. It has coded on the bottom these primers, these adapters. And they're complementary to these. So as it's flowing along, it'll stick to the adapters that are on the bottom of these lanes. Right? Is that clear so far? Okay. So it sticks to those. But you have single molecules of these kind of scattered all over this lane. And they don't really work very well. You have to increase the signal intensity because this is going to be a fluorescent signal that you're going to read. So you actually have to go what's called cluster growth. You have to increase the signal. And it actually is fairly simple. So you see this second adapter? It bends over and ligates or hybridizes to another region, and you just amplify. This is called bridge PCR, okay? And you get these clusters of all the same molecule, but they're in groups, scattered all over the inside of the lane. Now, you have this adapter, you have your sequence, and you have the open end of the primer sitting there, just like Sanger, right? So you can add a nucleotide at that position. The way that this works, it's a fundamental difference than Sanger, is that you add one nucleotide and it always terminates, it always stops. It stops at that position. So you add one nucleotide at a time. And the way it's done is that this is a flow cell, you just flow the nucleotides through. They'll add one base, you stop, and then you take a picture, okay, with a laser that will fluoresce each of those nucleotides at different wavelengths. And so you basically get a picture. It's a combined picture if you want to go through the details. But you get a picture with a lot of spots with different colors, right? After that picture is taken, this can actually be modified to unblock it. So you can let it extend another base. And then you flow nucleotides through again, and you add another base, and another base, and another base, and another base, and another base. So basically, you go through cycles where you are adding a base, taking pictures, unblocking, adding a base, taking pictures, unblocking, and so on. And each one of those, if you look at a spot, is a different color. Each spot corresponds to one sequence, right? So now, on one lane, you've got 200 million spots, each one of them corresponding to one sequence. That's how you generate 200 million sequences. Each one of these sequences is about 100 nucleotides. All right? So, yeah.
Sorry, so you can put all 4 nucleotides in at one time?
Yep. Each one has a
different colors?
Yep. Then they're all terminators at that point. But you can unblock them. That's the key difference with this and Sanger. Yeah.
And you're saying the fragments are approximately 100 nucleotides?
After you sequence, yeah. Yeah. You can technically keep sequencing them, but what happens is it's an artifact of the technology, is that the quality of the sequence will drop off and it becomes useless. But it's got resolution to 100 nucleotides.
And they anneal to the primers because those Y adapters
Yeah, so,
added to the fragments before.
well, the nucleotide is going to actually bind here, similar to Sanger. It's got a template that it's working on. In this case, the template is your sequence that you're working with. So it's going to add nucleotides to the sequence of that region. Any other questions?
So if they're only, so if they're only looking at that one spot that's circled, what's the deal with all the other colored spots? Like
Those are all actually these here. Those are all other sequences. So the software can actually look at each individual spot, keep track of all the different colors, and then at the end, it just counts up the sequence at the end. So basically, each one of these will correspond to a different sequence. Yeah.
And each one of those 100 base pairs, and then at the end, it compiles it into
Well, it gives you the raw sequence. Then you hand it over to people like us who have to do the analysis. That's
okay, but
yeah.
part of that analysis is finding the overlaps and actually looking to see.
Yeah. Yeah, and then you can use it, and I'll show you different applications you can use this for. So, yeah.
I might sound ridiculous, but
Yeah.
if it's all fragmented here,
Mhm.
how does it find its way back into
A genome, or
Yeah.
I'll talk about that in a
Okay.
second. Very briefly, though, but yeah, I'll talk about it. So,
Okay.
yeah.
Can I ask, are the fragments made randomly, like with physical stress, or is it restriction enzymes? Because I feel like you'd always miss a certain player.
Yeah, so actually there's different methods for fragmenting. It's a good question. So there are different methods for fragmenting and different ways to prepare libraries. It depends completely on the purpose of the experiment that you're doing. But in general, they use different instruments. The one I think Alvaro uses is a Cavaris, if I remember correctly. But he fragments basically to a specific size of about 200 to 400 bases. You can tune it so you get different size fragments. So you can create different types of libraries if you need to.
Then he runs another cell and selects a band.
Yeah, and he basically does. He runs a standard agarose gel, which you've probably done in classes or you've seen, and you separate based on size. And he will size select a specific size. He can cut it out and just pull and extract the DNA right from the gel.
But if you had 2 copies of the same genome, those 2 copies wouldn't fragment the same way every time.
Not necessarily, yeah.
Yeah.
I mean, it's going to be a technical issue there, but yeah. But basically, the idea is that if you have a consistent enough method for fragmenting, then you should see pretty comparable results.
We want it to be random.
Yeah, and you do want it to be random.
Because you don't want the same 100 bases stuck at the bottom of the
Yeah.
cluster every time.
Yeah, and actually, it's a good point to bring up, but This is one of the issues with actually Illumina. There are some biases. But in general, this tends to be quite useful. It's a random shearing of the DNA or whatever you're actually working on. So it works fairly well. Yeah?
Kind of interesting how, while the technology has moved forward, what you were just saying, is really old technology. The
Oh, yeah.
Augras and excising out and extracting that. So it's interesting how it really is a mix of the new and old.
Yeah, it's a basically this is a modification of Sanger, right? So it's not that far of a leap from what Sanger started. It's not extremely novel. Some of the newer generation sequencers actually are very different than this. But I don't talk about those because we didn't have the time. Okay, so I'm going to go ahead and move ahead if everyone's okay at this point. All right, so this is the big slide here. Sanger, okay, if you started sequencing a human genome now just using Sanger, you're probably talking about $300 million. 454 was the power sequencing that came out around 2005. I can't remember. It's been out for a little while now. It's actually obsolete. They don't exist anymore. I think they're basically done. But even then, it was pretty expensive. Illumina, you basically can take 2 lanes and get all the sequence that you want. 2 of those lanes out of that 8-lane slide, and you have a genome, right? You could do it in anywhere from 2 to 14 days, depending on what mode you run in. And you're probably looking at about $10,000 to get that sequence. There are some issues with it, but you basically get a random shearing and a coverage of your entire genome sequence. So you're getting kind of closer to that $1,000. Well, actually you are at $1,000 now. So Illumina released this thing, this monster, called the X10. This is basically 10 of those sequencers that you saw before, but optimized for human genomes. And they actually are now calling it the $1,000 human genome. So you're really already at a point where you can do this. And these are now being purchased at different genome centers all over the place.
So it's the same thing, but just 10 of them?
They've optimized it. They've done some tweaks to the technology that actually improve the sequence, improve the data that comes off of it, and it's geared towards human sequencing. So they actually don't even call it the human sequencing machine anymore. They call it the population sequencer. This can sequence 70 people in a week. Give you an idea.
But all it is 10 machines working together?
Yep. They're basically just 10 independent machines plugged in. They call it the X10, which I think is kind of funny, but.
Is the $1,000, like, what is the $1,000? Does the $1,000, is that, like, include recovery of the machines?
The $1,000 is going from here's my sample to here's the mutations that you have. It actually supposedly includes the analysis.
Okay.
I don't know how much I believe that. I'm on the analysis end, but basically that's supposed to be from everything to here are your mutations, and this is what we think diseases you might have or genetic predispositions for things.
What does the hardware cost?
The hardware, these are $500,000 a piece to $750. So times 10, you're probably looking at close to $5 million to $10 million. But I mean, $5 million to $10 million for companies that do this stuff, that's nothing. So yeah.
So how is it modified, or is the machines adapted for?
They basically, so the spots that you saw in the lanes before are fairly random, right? And you can imagine spots getting close together and mixing and causing problems. And so they actually set these up so they create little wells and they're separated. So you can actually get a really clean picture with as many spots as possible. So you increase the resolution and increase the quality of the sequence coming out.
So how is the human genome different than a fish genome?
Well, that's something you had to ask Illumina. We have those questions as well. They basically are focusing this towards the medical, because if you think about it from a sales point of view.
No, I totally understand.
Yeah.
But I'm just thinking,
Yeah.
I think in essence, it really is. It's a little fragments that you would run either way, and then
Yeah.
you just want to sell more.
Oh yeah, and I'm sure that if somebody's going to buy this and not use it for human, but that's what this is geared towards. And actually that includes the software. I mean, basically the idea is the software, everything that's in this is geared towards analyzing human data. It doesn't have fish genome in it, for instance. So, all right, so now actually the cost of the analysis, what you'll find now is the cost is really more on the analysis end than anything else. You could sequence quite a bit for about, I can't remember what Alvaro's prices are now. The library cost, the initial one was about $300, and a sequence of lane is about $3,000. But it's not that bad considering how much data you get out of it. And you can do all sorts of experiments on here, such as, let's say we work at an agricultural university and we got lots of different cool organisms like honeybee, or populations of organisms like Yellowstone, or, I mean, we could do all sorts of interesting things. So you could do genome assembly off of these. Genetic mutations, you could basically, you have a known human genome, and you want to find out what mutations a person might have that predispose them for a particular genetic disease. You can look at cancer. You can look at populations. So this is a technique they kind of call resequencing, because you already have a reference genome, but you're looking at a particular person and you're comparing the two. You also have, when you're looking at populations, these genome-wide association studies, or what are called GWAS studies. Assembling a genome, we talked about that. I think that question was raised. So at the very beginning, you shear the DNA, you create the libraries. This is a very, very, I would say, very simplified view of how it works. It's much, much more complex. But in general, you construct a graph based on the overlaps. It's a big jigsaw puzzle. And then you simplify this into a sequence. So you start with these fragments. A lot of these fragments will come together by the overlap, and you just collapse them into an assembly. It's more like this, right? Where you're taking a monster jigsaw puzzle and you're trying to put it together computationally. This is actually purely computational. You don't do this by hand. Mutations. So this is actually an example of the human genome. You can see this is a chromosome. You can see a centromere there at the top. That's a reference for chromosome, I think chromosome 22. And that's one base here. And these are all the next-gen sequences that are aligned against it. And the idea is you can look at those. This comes from a person. And you can say, what are the differences between the reference and that person? Right? And you can see in half the sequences, you get a change in a base, a C. Why is it in only half? Heterozygous, right? So this is a heterozygous mutation. So it's actually got the wild type allele in one and the alternate allele in the other chromosome. So you can do really interesting, cool things with this, including these big studies, these genome-wide association studies, where you look at migrations of modern humans. You can track changes because humans migrated out of East Africa, and you can follow, based on the patterns of changes, migration paths, right? So you can do some very interesting, cool things. The Thousand Genomes Project actually really fed into this. Other applications, which I won't go into any detail out because we're probably running close out of time. No, we're okay. RNA Seek. So you care about gene expression. What genes are turned on and turned off in different experimental conditions, right? So you want to say treat something and to see if the gene is turned on. You can use this technology and actually get at that information. Epigenetic effects. So how many people know what epigenetics is? It's Yeah, so I'm sure they talked about it. Basically, this can look at transcription factor bindings of binding sites. Most transcription factors bind to specific regions of DNA. Well, you can sequence that DNA, right? You just shear it. So the idea is that you sequence only those things the transcription factors bind to. Histone modification. This can actually look at, and actually it's developed primarily for histone modification. Microbiome analysis. So, which she works on. So looking at what organisms are present in a particular sample. Gut, Yellowstone, soil. I mean, basically looking at what is there. So you can apply this to tons of different things if you think about it. It's a very powerful technology. And so I'm going to go through a few examples of what it actually has led to. And you'll see that a lot of these are really current examples. This one was a story that actually came out in the New York Times. It's been out for a little while now, but they found some interesting things that have led from the initial study. So this is an enzyme deficiency called NGLI1. And mutations in this particular enzyme are known to cause problems with movement, development delays, liver problems, but they have an interesting odd symptom in that they don't shed tears. They will cry and they don't shed tears. This actually happens in newborn infants going up to about the age of 2 or 3. It is a particularly terrifying disorder because parents who have a child that has this, they have no idea what's going on because it's extremely rare. There are no known cases up until a couple of years ago that this even existed. Okay. The enzyme that causes this just basically takes these defective, they call it defective products, but it's an end product in a pathway, a biochemical pathway, and recycles them. It gets rid of them. If those aren't recycled, it builds up and it causes these issues. So the whole impetus actually came from, this is a blog, as a snapshot. I like the Liam Neeson there. Matthew Might actually wrote this blog. He had his son, who's actually still alive. This is a little bit misleading. But basically, he had a son that exhibited all these symptoms. And they could not figure out what was going on until they actually went to a doctor who proposed, well, why don't you try sequencing to see if you can figure out or work out what particular genetic change may be causing this. And they linked it back to this one particular NGLI1 gene. It's a genetic link. It's recessive. You have to have 2 copies of it. That's why it's extremely rare. And at that point in time, this was the only case known. He actually thought it was unique. So he put this blog up, detailed it, talked about the symptoms and everything. And what actually they found out was that there were 8 more cases of this out there where parents didn't know, right, that their child had this. So it was a very good thing. They actually were able to track down what the mutation was and actually try to find some solution to it. At this point, they actually are just treating the disease and they don't have an actual cure or a long-term prognosis. But it's a bit more positive because they can start working towards things. This was a story in CNN that actually is one of the other patients, this girl, Grace Wilsey. That's from March. This is past March. So very recent. Sequencing to find a cause of a disease. A boy in Wisconsin was in a hospital, was in a coma. They didn't know what was causing it. They basically used a new approach where they took his cerebral spinal fluid because he had brain swelling. They thought, well, something may be in there that's causing this. Took that and said, let's just see if there's anything in there that could be causing this disorder or this disease. And they found a cause within 48 hours. It was leptospira Spira? Leptospira. I can never get that correct one. He was swimming, basically, and I think they had sewage contamination. They think that it might have been something that came from a particular swimming hole. But the thing is that it's treatable, and they treated him with antibiotics, wiped it out within days, and there he is. Okay? The interesting thing about this is that, so this is from June, just a month ago. So Joe DeRisi, this researcher here, wrote up this bioinformatics pipeline. He basically says, well, you know, if you want to do this, or we're just going to make it available. You could put this up, and anybody could use this particular resource. We're going to make it available. Actually, I think he put it up on Amazon. They have an Amazon Web Instance where you can make this, where you can do this work. But you just have to have the DNA sequence, feed it through this pipeline, and it will say, this is the possible pathogen that's in your sample, right? And he did it for free, by the way. This is all published, but you basically, you just got to pay for Amazon to do the analysis, but it's not that bad. It's pretty cool. You're probably going to hear a lot about Joe DeRisi in the next 5 years or so. Some more controversial uses. This is a population study that was done in Arizona. There was a group of Indians that gave blood samples to geneticists at the university there to try and find the cause. They have a high rate of diabetes in this particular tribe. The researchers instead turned around and published theories about mental illness, the geographic origins of the tribe, inbreeding in the tribe, and then they didn't find out about this until one of the people in the tribe went to the defense for the student. And it came up that they were doing all this other work. And by the way, I don't see diabetes in there, right? So the tribe members stood up at the end of the defense and said, did you get our consent to do this? And they stopped the defense. They went through the courts. The courts basically gave them all of their samples back. And wet restitution on top of that. But the thing is that, you know, their sequence is kind of floating out there. This is actually one of the older cases. This is right around the time when these technologies started coming out. That's just 2010. It's not that long ago that this happened. Sequencing your son for fun. This was one that just happened out of California. A researcher wanted to have his son's genome sequenced. He took a sample from the placenta of his unborn child and sequenced it. So this is a researcher, so he had access to resources and did it. He actually said it was more cool to have it than practical. And he actually wrote this on his blog. He said, deal with it. This is going to happen, right? So did his son have any say in this? No. Did his family have any say in this? Because he's not just sequencing his son's DNA, he's sequencing his mother's, part of his mother's chromosome, right? His mother's genetic information, part of his family's genetic information. It's all in there. So that's something that he never even considered. He just did it. That's from June. And of course, I don't know if anybody's seen this. It's a great book, right? Henrietta Lacks. So the book itself doesn't really talk about the genome sequence, but there's a second story to this in that when this book was going through the publication phase, some researchers in Germany, and I think some researchers actually in the States, took the HeLa cells and sequenced the genome. And then they were publishing it. And do you think that they asked the family? They didn't actually. The family was very gracious about it. NIH contacted them, said that this is coming down the pipeline. They halted the publication. They said that, you know, we got to make sure that they have consent. They actually stopped it and dealt with it in a meaningful period of time, but it still made national news. The family basically said, look, it's already out there. The cells have been out there for a long time. We need to make it publicly available. It's great for research, but they should have been contacted. That was from this past fall. And then, of course, there's the plain weird, which I wanted to end this on. And that is the Sasquatch Genome Project. Although I still say if you've got a stock photo of tubes on your front page, that probably causes problems with your credibility. Also, and the other thing about this is they actually created their own journal. It's the only publication in this particular journal, by the way. They don't have any other publications in it. So I think that kind of affects their credibility just a tad. There might be a little bit of doubt. All right, so sequencing has come a long way, and it's been 40 years, roughly, about 40 years since the technology really started up. What took decades and billions of dollars now takes days and thousands, and it's getting cheaper. And it basically has opened up a huge field in omics to the point that actually Jonathan Eisen has now coined what he calls the Badomics Award, because people just make up these catchy, flash in the pan phrases. So if you ever want to see some people overselling or trying to get grants, this is a good place to go. He tags them pretty well. And really, the era of personal genomics, they keep saying, well, it's a little ways off. No, it's here. It's already started. They already got the sequencers there. It's just going to take a little bit more time. So medications cost $1,000. They're covered by insurance. Why not sequencing? Question. So does anybody here want their genome sequence given to their insurance company? So we're still wrangling with all these kind of ethical things. What do we do with all this information? So that's it. I'm just going to tell, I got a lot of this information from Victor, Radhika, Luda, Alvaro, and Chris for the sequencing information. But does anybody have any additional questions on this? Be glad to answer anything. So, yep.
So when you do the next generation sequencing, any sequence you get is of one haplotype at a time.
Yeah, in general.
So you don't have to deal with like the double peaks that you would see in Sanger.
Right.
They're just because
they're basically single molecule.
Okay.
You get some amplification there, but you saw that it starts from a single molecule.
Okay.
So yeah. So basically, that's one of the reasons why it's very powerful for this. Whereas with Sanger, you're actually doing everything in one well.
Yeah.
So you can get mixes depending on how you prep the DNA. But yeah.
Like, how much of an issue is it though when you start to get length differences between chromosome you got from mom and chromosome you got from dad?
Oh, so differences so deletions and insertions. Yeah. You can actually detect those to some extent. There are some barriers for this technology. It's short reads. You're still at 100 nucleotides. A Sanger read is 500 to 700, depending on how you sequence it and how clean the DNA is. It could be a little bit more, but you'd still start from a single molecule, and the reads, when they first started this technology, the reads were 25 nucleotides, and this summer they're actually going to 250. Sorry, so they're just getting longer,
Yeah.
and now they have single molecule technology. PacBio, or Pacific Biosciences, has technology where they can read a single molecule through a pore and then get the sequence from that. And that's 10,000 bases on average, 5 to 10,000. The thing about those is that there's a problem with error rate. So you wouldn't want to use those for looking at mutations. But you could probably look, large-scale deletions if something's missing or something's inserted. That would probably give you a pretty good idea of what's there. And and that's only improving as well. So that's the next step.
If you were trying to do a human genome, how often do you pick up like viral or bacterial DNA?
All the time.
Or blood or something?
I'll cloak it in abstracts. So I'm working on a project where somebody has done work on human brain, and they were looking for basically somewhat like what Joe DeRisi does, but at a much, it's a needle in a haystack. Basically, at a much finer scale, you're looking for viral causes of specific genetic, or not genetic, but neural disorders. So they're looking at the brain. A region of the brain, they take that, they sequence it, and they basically look for anything that's not human, right?
So you have to get everything and then filter it out.
Yeah, and you see things, you can get viral traces in there. We've seen lots of interesting things. We've seen actually they have a lab artifact in their lab. They have a mouse virus that they know as a contaminant, and we see it in the sequence data. It's that clean. It's pretty interesting. Yeah.
How do they decipher between the different chromosomes? If you're just doing all the DNA, is that just something that they know?
So the general procedure for doing, if you're looking at human changes, is you already have a human reference, and those are actually based on chromosomes. So you basically will take the sequence that's from a person, and you take it and you align it to the reference sequence. So you're trying to actually, and that's a good thing because you're trying to get differences between them.
So that original reference may have been like that original human genome project.
Yeah, and
$1 billion.
Yep.
That they would have maybe separated and done by chromosome.
Yep.
Is that true? Okay.
Well, it's a little bit more technical. They scaffolded it. They basically said, well, we know that this is where these pieces go based on genetic mapping, based on other pieces of evidence. They just basically lay it out on a chromosomal basis.
So that's already been done, and then they can just match up.
Yeah,
Okay.
that's basically it. Yeah, yeah.
For preserved human remains, so find 1,000-year-old mummies and so forth, can you sequence genomes for those remains?
Depends on it depends on the quality of the DNA that comes out, but they've done this.
Okay, so then when they track migration of people out of Africa and into other continents, are they using any of that information, or can they just strictly use modern?
No, with the 1,000 Genomes project, they're using modern, but they have gone back and extracted sequence from older samples. Actually, I think one of the recent ones was, well, this is really more for a disease, Black Plague, so Yersinia. They were able to reconstruct the original sequence of the plague genome by going back and sequencing samples of people who died at that point. They're buried in mass graves. They're out there, and so they could actually get that information from there. They did the swine flu, I think, was another one. So yeah, that's the thing about it is you can see kind of the technology really opens up a lot of things. Yeah, but you can think about some of the implications there as well. Yeah.
So I know obviously this is super new and laws are slow to come
Yeah.
on the heels, but I guess I don't really know what's out there, or what are your thoughts on kind of where, I mean, because I'm thinking like we're going to be in Gattaca, like, I mean, that movie's 15 years old, but like one day it's going to be like that where we're going to have designer babies, right? I mean, I'm being a little facetious, but like that's almost kind of where it could go if there's no.
I think there are people, so with current sequencing, they're really working through some of the ethical issues that are coming up. So for instance, with samples, even with the human microbiome, okay, so you have a sample that comes from gut, and you're looking for microbial samples, but actually you get a lot of the person's DNA in there as well. So you can identify who that person technically is and what their gut sample is by looking at that sequence. They anonymize the data. They basically say this goes to patient X. And they're actually trying to find ways of not just anonymizing it, but actually dealing with the sequence data so that you can't detect the human component of it at all. That's really tricky, by the way. So you can't. It's not an easy problem to solve. For human data, they basically have it behind a locked door, and you have to ask for access to it through what's called, it's DB gap. But you can get access to certain data if you ask for and go through the proper channels. That way they know who you are and know who has access to what. Yeah.
I'm remembering reading something in Science, I don't remember where I read it, of where people have actually gone back, sort of a proof of concept really, to show that they can in fact identify people out of published data.
23andMe, actually, that some people were able to do it through 23andMe. They did it through, I think they did some off of the 1,000 Genomes project, and they did something through 23andMe. They were able to, so you have genealogy databases that are out there that have all sorts of phenotypic information in it. So this, if you come from a population that has a particular genetic predisposition for a disease, or they have a phenotypic characteristic, or they are located in a particular geographic location, then you can sort of act as a detective and trace your way back and try to figure out who that person was. And they were able to figure out, or work out, I think 10 people, I can't remember how many it was from the 1,000 Genomes project, based just on the genetic information. So there's only so much you can anonymize, but then the task of doing all that is, now, that doesn't mean somebody's not going to create some software to do it for you in the future. That could happen, but you have to have a database there to start with. I think it's, yeah.
I've seen these advertisements on TV for Ancestry.com, this new DNA testing.
Yeah.
I don't know how you do it, but
Yeah.
is that valid? And where is the database? What's the database there?
Well, you would have to check through it. I would probably think about 23andMe, but I don't know if I'd ever do my sequence. So 23andMe doesn't use sequence. They use what's called a SNP array. But they are going to sequence at some point. Ancestry is sequencing. And they're creating a database, like a genealogy database, where you can trace back origins. So if you're from a large family and you found your sister was a half-sister, then those things are easily possible now. And so do you want to know that information? I mean,
It just seems so expensive for a service like that.
you can cut the cost down. And my guess is that, so the cost for 23andMe is like $90. I think it was $90 or $99 or something like that. And that might be what Ancestry is doing. It's very similar to that. I don't know how successful they are, but I can tell you it's not that far from sequencing. If it's $1,000, it's only going to get cheaper over time. So it's, sort of heading in that direction.